Hey, genuine well-wishers and followers, how are you, good-hearted fellows? It's ideally Earthling Professor S. Suresh with absolute delight back for my 56th video in pipeline for publication on YouTube today, 4th of August 2020, Tuesday. Topic for discussion today is vaccine development phases and time frame for marketing. This is appropriate time for such a topic. A vaccine is a biological preparation that provides action-required immunity, acquired immunity to a particular infectious disease. A vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of the microbe, its toxins, or one of its surface proteins. The agent stimulates the body's immune system to recognize the agent as a threat, destroy it, and further recognize and destroy any of the microorganisms associated with that agent that it may encounter in the future. Vaccines may be prophylactic or therapeutic. The administration of vaccines is called vaccination. Vaccination is the most effective method of preventing infectious diseases. Widespread immunity due to vaccination is largely responsible for the worldwide eradication of smallpox and the restriction of diseases such as polio, measles, and tetanus from much of the world. The effectiveness of vaccination has been widely studied and verified. The World Health Organization, WHO, reports the licensed vaccines are currently available for 25 different preventable infections. The term vaccine and vaccinations are derived from variole vaccinae, smallpox of the cow. The term derived by Edward Jenner to denote cowpox. He used it in 1798 in the long title of his inquiry into the variole vaccinae known as the cowpox, in which he described the protective effect of the cowpox against smallpox. In 1881, to honor Jenner, Louis Pasteur proposed that the terms should be extended to cover the new protective inoculations then being developed. There is overwhelming scientific consensus that vaccines are a very safe and effective way to fight and eradicate infectious diseases. Limitations to their effectiveness nevertheless exist. Sometimes the protection fails because of vaccine-related failures, such as failures in vaccine attenuation, vaccination regimes, or administration, or host-related failure due to host immune system simply does not respond adequately or at all. Lack of response commonly results from genetics, immune status, age, health, or nutrition status. It also might fail for genetic reasons if the host immune system restraints includes no strains of B cells that can generate antibodies suited to reacting effectively and binding to the antigen associated with the pathogen. Even if the host does develop antibodies, protection might not be adequate. Immunity might develop too slowly to be effective in time. The antibodies might not disable the pathogen completely, or there may be multiple strains of the pathogen, not all of which are equally susceptible to the immune reaction. Adjuvants commonly are used to boost immune response, particularly for older people, 50 to 75 years and up, whose immune response to a simple vaccine may have weakened. The efficacy or performance of the vaccine is dependent on a number of factors: the disease itself, the strain of the vaccine, whether the vaccination schedule has been properly observed, idiosyncratic response to vaccination. Some individuals are non-responders to certain vaccines. Assorted factors such as ethnicity, age, or genetic predisposition. If a vaccinated individual does develop the disease vaccinated against, the disease is likely to be less virulent than in unvaccinated victims. The following are important in effectiveness of the vaccination program: careful modeling to anticipate the effect that an immunization campaign will have on the epidemiology of the disease in the medium to long term; ongoing surveillance for the relevant disease following introduction of a new vaccine; maintenance of high immunization rates even when a disease has become rare. Vaccines led to the eradication of smallpox, etc. Other diseases such as rubella, polio, measles, mumps, chickenpox, and typhoid are nowhere near as common as they were 100 years ago. As long as a vast majority of people are vaccinated, more than 50% of the population, it is much more difficult for an outbreak of disease to occur, let alone spread. This effect is called herd immunity. Polio, which is transmitted only between humans, is targeted by an extensive eradication campaign that has seen endemic polio restricted to only parts of 3 countries: Afghanistan, Nigeria and Pakistan. Vaccination programs have greatly reduced the prevalence of infections resistant to penicillin or other first-time antibiotics. The measles vaccine is estimated to prevent 1 million deaths every year. Vaccination given during childhood is generally safe. Adverse effects, if any, are generally mild. The rate of side effects depends on the vaccine in question. Some common side effects include fever, pain around the injection site and muscle aches. Additionally, some individuals may be allergic to ingredients in the vaccine. At least 19 countries have no-fault compensation programs to provide compensation for those suffering severe adverse effect of vaccination. Vaccines contain dead or inactivated organisms or purified products derived from them. There are several types of vaccines in use. Inactivated vaccine is one, but generally virulent microorganisms that have been destroyed with chemicals, heat or radiation. Examples include polio vaccination, rabies vaccines, and some influenza vaccine. Some vaccines contain live attenuated microorganisms. Many of these are active viruses that have been cultivated under conditions that disable their virulent properties that use closely related but less dangerous organisms to produce a broad immune response. Although most attenuated vaccines are viral, some are bacterial in nature. These typically provoke more durable immunological responses and are the preferred type for healthy adults. They may not be safe for use in immune compromised individuals and on rare occasions mutate to a virulent form and cause disease. Toxoid vaccines are made from inactivated toxic compounds that cause illness rather than the microorganisms. Toxoid vaccines are known for their efficacy. Not all toxoids are for microorganisms. Rather than introducing an inactivated or attenuated microorganisms to an immune system, a subunit vaccine uses a fragment of it to create an immune response. Like hepatitis B virus is a subunit vaccine. The virus-like particle vaccine against human papillomavirus, shortly HPV, that is composed of the viral major capsid protein and the hemagglutinin and neuraminidase subunits of the influenza virus. The subunit vaccine is being used for plague immunization. Certain bacteria have polysaccharide outer coats that are purely immunogenic. By linking these outer coats to proteins, the immune system can lead to recognize the polysaccharide as if it were a protein antigen. This approach is called is used in the Haemophilus influenza type B vaccine. These are called conjugate vaccines. Heterotypic vaccine, also known as heterologous or Jenerian vaccines, are vaccines that are pathogens of other animals that either do not cause disease or cause mild disease in the organism being treated. While most vaccines are created using inactivated or attenuated compounds from microorganisms, synthetic vaccines are composed mainly of synthetic peptides, carbohydrates, or antigens. Vaccines may be monovalent, also called univalent, or multivalent, also called polyvalent. A monovalent vaccine is designed to immunize against a single antigen or single microorganism. A multivalent or polyvalent vaccine is designed to immunize against two or more strains of the same microorganism or against two or more microorganisms. The valency of the multivalent vaccine may be denoted with a Greek or Latin prefix, tetravalent, quadrivalent, etc. In certain cases, monovalent vaccine may be preferable for rapidly developing a strong immune response. The immune response system recognizes vaccine agents as foreign, destroys them, and remembers them. When the virulent version of an agent is encountered, the body recognizes a protein coat on the virus and thus is prepared to respond by first neutralizing the target agent before it can enter the cells, and secondly by recognizing and destroying infected cells before that agent can multiply to vast numbers. When two or more vaccines are mixed together in the same formulation, the two vaccines can interfere. This most frequently occurs with live attenuated vaccines, where one of the vaccine components is more robust than the others and suppresses the growth and immune response to the other components. Vaccines typically contain one or more adjuvants used to boost the immune response. Vaccines may also contain preservatives to prevent contamination with the bacteria or fungi. Until recent years, the preservative thiomersal was used in many vaccines that did not contain live virus. In order to provide the best protection, children are recommended to receive vaccinations as soon as immune systems are sufficiently developed in respond to particular vaccines, with additional booster shots often required to achieve full immunity. This has led to the development of complex vaccination schedules. In order to combat declining compliance rates, various notification systems have been instituted and a number of combination injections are now marketed. Besides recommendations for infant vaccinations and boosters, many specific vaccines are recommended for other ages or for repeated injections throughout life, most commonly for measles, tetanus, influenza, and pneumonia. Pregnant women are often screened for continued resistance to rubella. The HPV is recommended in the US as of 2011 and UK as of 2009. Vaccine recommendations for the elderly concentrate on pneumonia and influenza, which are more deadly to that group. In 2006, a vaccine was introduced against shingles, a disease caused by the chickenpox virus, which usually affects the elderly. First generation vaccines are whole organism vaccines, either live or weakened or kill form. Second generation vaccines were developed to reduce the risks from live vaccines. These are subunit vaccines consisting of specific protein antigens. They can generate antibody responses but not killer T cell responses. DNA vaccines are third generation vaccines. They contain DNA that codes for specific proteins, antigens from a pathogen. The DNA is injected into the body and taken up by cells whose normal metabolic processes synthesizes proteins based on the genetic code in the plasmid that they have taken up. Because these proteins contain regions of amino acid sequences that are characteristic of bacteria or viruses, they are recognized as foreign, and when they are processed by the host cells and displayed on the surface, the immune system is alerted, which then triggers immune responses. Alternatively, the DNA may be encapsulated in protein to facilitate cell entry. In this capsid protein, if this capsid protein is included in the DNA, the resulting vaccine can combine the potency of a live vaccine without reversion risks. One challenge in vaccine development is economics. Many of the diseases most demanding vaccine, including HIV, malaria, and tuberculosis, exist primarily in poor countries. Pharmaceutical firms and biotechnology companies have little incentive to develop vaccines for these diseases because there is little revenue potential. Even in more affluent countries, financial returns are usually minimal and the financial and other risks are great. Most vaccine development to date relied on push funding by government, universities, and non-profit organizations. Many vaccines have been highly cost-effective and beneficial for public health. The number of vaccines actually administered has risen dramatically in recent decades. This increase, particularly in the number of different vaccines administered to children before entry into schools, may be due to governmental mandates and support rather than economic incentive. Lifestyle diseases are also on the increase. Zoonotic diseases are also in the increase because our lifestyle has changed. People are becoming more sedentary, eating anything that moves. All these are the reasons. According to WHO, the biggest barrier to local vaccine production in less developed countries has not been patents, but the substantial financial infrastructure and workforce expertise requirements needed for market entry. There are no true genetic vaccines. The vaccine produced by a new facility must undergo complete clinical testing for safety and efficacy similar to that undergone by that produced by the original manufacturer. For most vaccines, specific processes have been patented. These can be circumvented by alternative manufacturing methods, but these require R&D, infrastructure, and a suitably skilled workforce. In the case of a few relatively new vaccines such as HPV, the patents may impose an additional barrier. Vaccine production has several stages. First, the antigen itself is generated. Viruses are grown either on primary cells such as chicken eggs or on continuous cell lines such as the cultured human cells. Bacteria are grown in bioreactors. Likewise, the recombinant protein derived from the viruses or bacteria can be generated in yeast, bacteria, or cell cultures. After the antigen is generated, it is isolated from the cells used to generate it. A virus may need to be activated, possibly with no further purification required. Recombinant recombinant proteins used need for many operations involving ultrafiltration and column chromatography. Finally, the vaccine is formulated by adding adjuvant, stabilizers, etc. And preservatives as needed. The adjuvant enhances the immune response of the antigen, stabilizers increase the storage life, and preservatives allow the use of multi-dose vials. Combination vaccines are ordered, developed and produced because of potential incompatibilities and interactions among the antigens and other ingredients involved. In 2010, India produced 60% of the world's vaccine worth about $900 million. Many vaccines need preservatives to prevent serious adverse effects such as staphylococcus infection, which in one 1928 incident killed 12 out of 21 children inoculated with a diphtheria vaccine that lacked the preservative. Several preservatives are available, including thiomersal, phenoxyethanol, and formaldehyde. Thiomersal is more effective against bacteria, has a better shelf life, and improves vaccine stability, potency, and safety. But in US and the other countries, EU, it is no longer used for childhood vaccines as a precautionary measure due to its mercury content. Although controversial claims have been made to that thiomersal contributes to autism, no convincing scientific evidence supports these claims. Further, MMR vaccine does not cause autism and actually reduce the risk of autism by 7% as per a study. The final stage in vaccine manufacture before distribution is fill and finish, which is the process of filling vials with the vaccines and packaging them for distribution. Although this is a conceptually simple part of the vaccine manufacture process, it is often a bottleneck in the process of distributing and administering vaccines. Notable developments in vaccine delivery technologies have included oral vaccines. The microneedle approach, which is still in stages of development, uses pointed projections fabricated into arrays that can create vaccine delivery pathways through the skin. An experimental needle-free vaccine delivery system is undergoing animal testing. A stamp-size patch similar to the adhesive bandage contains about 20,000 microscopic projections per square centimeter. This dermal administration potentially increases the effectiveness of vaccination while requiring less vaccine than injection. The use of plasmids has been validated in preclinical studies such as a protective vaccine strategy for cancer and other infectious diseases. However, in human studies, this approach failed miserably. The overall efficacy of plasmid DNA immunization depends on increasing the plasmid's immunogenicity, while also correcting the factors involved in the specific activation of immunocollector cells. Vaccine development has several trends. Until recently, most vaccines were aimed at infants and children, but adolescents and adults are increasingly being targeted. Combinations of vaccines are becoming more common. New methods of administering vaccines are being developed, such as I mentioned before, even aerosol via inhalation devices, and eating genetically engineered plants. Vaccines are being designed to stimulate innate response. Attempts are being made to develop vaccines to help cure chronic infections as opposed to preventing disease. Vaccines are being developed and defend against bioterrorist attacks such as anthrax, plague, and smallpox. Appreciation for sex and pregnancy differences in vaccine responses might change the strategies used by the public health officials. Scientists are now trying to develop a synthetic vaccines by reconstructing the outside structure of a virus. This will also help prevent vaccine resistance. The development, testing, and regulation of vaccines evolved to the current system in the 1900s. Development can take many years, and if it succeeds, it could end up to a large-scale vaccination program. Many of these vaccines fail at the beginning because they are unsuccessful to produce the desired immune response. Only a few of the possible vaccines make it to the licensing. Development of every vaccine starts with the exploratory stage It consists of laboratory research to identify antigens that might be able to treat or prevent a disease. The found microorganisms are weakened or killed so that they cannot replicate and cause a disease anymore. The wanted part of the microorganism is used to manufacture a vaccine. In the EU, development of vaccines can be divided into two stages: preclinical and clinical development. Preclinical studies include laboratory and animal studies. Once the wanted microorganism has been found and antigens isolated. Poor animals. Guinea pigs. The purpose of pre-testing is to assess safety, biological activity and formulations. Now it is banned actually. Okay, animal studies can also help to see what kind of cellular responses is expected in humans. Good manufacturing practice standards are used when the cell line is constructed and cell banking is done. In the stage of clinical development, the vaccine is tested in humans. The testing happens in 4 phases and can take many years. First 3 phases are clinical trials. The stage of clinical development is based on strict ethical principles. Volunteers are well informed of the vaccine's safety and efficacy. Phase 1 is done in small scale with 20 to 100 healthy volunteers. Its purpose is to determine safety and dosage. Vaccines that are targeted to children are first tested with adults. Volunteers in these studies are attentively monitored and conditions are controlled. Phase 2 is tested with a larger group of 100 to 500 patient volunteers. The purpose of this phase is to evaluate the effectiveness of the vaccine versus clinical diseases and artificial infections. Immune response alongside side effects is also evaluated and the method of the delivery is decided. Phase 3 confirms effectiveness and monitors adverse effects events originating from long-term use. This phase is tested with 1,000 to 5,000 patient volunteers to evaluate the effectiveness under natural disease conditions. The tests in this phase are randomized and the developed vaccine is tested against a placebo. If the vaccine is safe and effective for a defined period, the manufacturer can apply for a license from the regulatory authorities. If the license is admitted, can the manufacturer start to market the vaccine for human use? In phase 4, the vaccine has been licensed and is available for the users. The vaccine is still under being surveillance and the aim is to detect adverse events. The effectiveness of the vaccine is monitored throughout its long-term use. Process for manufacturing a biological preparation consists of upstream and downstream processes. Upstream includes the entire process from early cell isolation and cultivation to cell banking to the culture expansion of the cells, fermentation process and the final harvest. Downstream involves separation of the cells from the media, isolation of the product, purifying, polishing and packaging of the product. In short, it is where the capturing of the desired protein happens. Harvesting happens between upstream and downstream process. The pharmaceutical industry is one of the most regulated industry known. Governments in all over the world have established regulatory agencies to ensure that every aspect of pharmaceutical activity is tightly controlled. The WHO does the recommendations for biological products that are used internationally. Many countries follow the WHO standards. National regulatory authorities, NRAs, are agencies which ensure that the pharmaceuticals and biological products that goes to the market meets the international expectations of quality and safety. All countries that produce vaccines must follow 6 critical control functions. These functions are a published set of requirements for licensing, surveillance of vaccine field performance, system of lot release, use of laboratory when needed, regular inspections for good manufacturing practices and evaluation of clinical performance. Recombinant vaccines are the new kind of vaccines developed with the new recombinant DNA technology. It's a technique where genes from one organism are isolated, purified and then reproduced in another organism, and it is often performed through ligation. Vaccines manufactured by using a recombinant DNA technology are safe and production costs are lower compared to traditional vaccines. Vaccines made by this technology can be divided to DNA vaccines and recombinant protein subunit vaccine. Some vaccines need a booster shot to maintain the protection against diseases. This kind of shot is called homologous prime boost because it uses same formulation. Vaccine manufacturing is a long and expensive process. Vaccines are proven to have important influence in the human health. The field of vaccines is evolving rapidly and new findings are constantly made. The future of vaccines depends on the successful medical research and new vaccine technologies for the development. Debates and discussion on topics like this can go on forever. Let me call it a day. We'll meet again real soon. Some of the statements views expressed are solely mine based on limited knowledge gained over 6 decades. Always watch these videos with closed captions, subtitles for absolute 100% comprehension as per me. Sincere recommendation zillion times so far. I know I rushed through to complete the presentation within 20 minutes max. This is the time limit I had set myself to retain the viewer's attention as of now. I know nowadays I exceed this time limit since I make honest attempts to cover the selected topic more elaborately, including so many intricate details, referring a wide variety of sources, in particular Wikipedia quite extensively, besides articles and books published recently. In addition, I add my own salt and pepper and masala based on my personal experiences so far in my life and also intuition. Stay at home as much as possible to maintain social distancing, attributed to pandemic, novel COVID-19, and prolong your lifespan. Lead a healthy life. God alone can put a stop to natural and unnatural events, maybe bio-war, cyber-war, like pandemics, which may lead to apocalypse and extinction of all sentient species on earth, for no fault of theirs. All the best. Rest in peace and harmony.